Today we shall study the second type of immunity, that is specific immunity. In specific immunity there are two types of cells which are involved in specific immunity. These are called as T lymphocytes and B lymphocytes. T lymphocytes get matured in the thymus gland, therefore they are called as T lymphocytes. They are involved in a type of immunity which is called as cell mediated response. So what does happen in cell mediated response? In cell mediated response, the cells themselves, they are involved in the destruction of antigens. So firstly, you should be able to differentiate between antigens and antibodies. Antigen is any substance, protein or polysaccharide which can stimulate the production of antibodies is called as antigen. Antigens are usually foreign or non-self substances, and the antibodies are produced by our immune cells. So in this case the antibodies will be present on the surface of T cells. When the antigen will come, this black block is an antigen. It will be recognized by the antibodies which are present on the surface, and next, after being attached on the surface, they will be ultimately destroyed by phagocytosis, while some of these cells will be converted into plasma cells. Plasma cells are the cells which are involved in the production of antibodies. These antibodies can be free or these antibodies can be attached as well. So these antibodies will be produced and they will be simply released into the body. So next time in case of attack, what will happen, these antibodies will already be present in a huge amount, so that the antigen can be killed effectively. You can see the antibodies were already present on the cells. Next type of response is called, in case of specific immunity, is called as humoral immune response. In case of humoral immune response, there is antibody production, free antibody production. The cells which are involved are called as B cells. What does happen? These are the free antibodies which are present in our blood, and they will be binding with this antigen surface, so that this antigen can be exposed to the phagocytic cells. This is the phagocytic cells. When this antibody will bind, this will help this antigen to bind on the phagocyte, on the surface of phagocytic cell. You can see. So what will happen after this? Obviously the antigen will be destroyed, while next these cells will be converted into two types of cells. There can be two fates of these cells. Either they can be converted into memory cells. Memory cells are a type of clone cells. They will be similar to these phagocytic cells, but they will be having some genes of this antigen, so that if in case of a large number of antigens entering into our body during some later stage of life, this antigen is present into their cells, and these cells will be dividing and they will be producing antibodies in a large number. Others will be converted into plasma cells. Plasma cells will be producing free antibodies into the blood, so that the antigen can be trapped. So this was a type of specific immunity. In both of these types of immunity, you can see that the antigens and antibodies interactions are very specific. The receptors present on the surface of antigen or the shape of antigen is recognized by specific antibodies, and then they are killed. Means as you can see, there was a block shaped receptor, and if it was of some other shape, like it was circular or it was triangular, the cells would not have been able to recognize and then destroy it. So this was the specific immunity. Next we have the antibody. As we are continuously discussing antibody. So these antibodies are actually immunoglobulins. Globulins means they will be globular proteins, and immuno means which the proteins which are involved in immunity. As you can see, these antibodies are having a structure of 4 polypeptide chains. There are 4 polypeptide chains, 2 lighter chains, this one, and 2 heavier chains. These are the heavy chains. In all of these chains there is a region which is called as constant region, and it is same in all of the antibodies, while this region which I have marked black, this is called as variable region. It is specific in every antibody, and it is the region which helps the antibodies to attach with the antigens specifically. Its structure will be different. The sequence of amino acids in this region, in this variable region, will be different in case of each antibody, so that it can bind specifically with the antigens. These antibodies are obviously the major component of our immunity. Next is the type of immunity. Immunity is of 2 types: active immunity and the passive immunity. Active, as its name depicts, it's mean the cells will be, the body will be using its active energy resources. It will be using its own energy to produce antibodies or to kill the cells, mostly antibodies. It is either natural or it can either be artificial. In case of natural immunity, like this was the specific immunity, either cell mediated or the humoral immune response, the pathogen first enters, it first affects the body, and then the cells get to know how to synthesize antibodies and what can be the shape of antigens, so they can synthesize antibodies accordingly. Like we have if we have flu, you must have read about, or you must have heard about the plasma donation nowadays related to corona. So first coronavirus enters into the body. Next time our body cells will be having a large number of antibodies to cope up with the coronavirus. So yeah, the blood of affected patients is having anti-coronavirus antibodies. So this anti-coronavirus antibodies containing blood can be used to treat the patients who are critically who are in a critical condition. So next is the type of artificial immunity. In case of artificial immunity, the cells are artificially induced to produce antibodies. It is vaccination actually. In this case, weak pathogens will be added. Doesn't matter if it is strong, it is virulent, it is causing disease, or it is not causing disease. Mostly its shape is recognized by ourselves. So in case of artificial immunity, the pathogen which is entered into our body will not be able to cause disease. Mostly it will be either heat killed or weakened by some method, and although it is weakened, but it will be able to induce our cells to produce antibodies. So it will produce immunity into our cells, like we are vaccinated against polio, in which weakened germs or some parts of the polio virus which are not able to induce polio, they are inducted into our body, and our body gets immune to poliovirus. Even if the strong virus or the virulent virus enters into our body, it will not be able to cause disease. Next is the passive immunity. As I have told you about the plasma therapy, in case of passive immunity, usually in case of um, extremely critical condition, the patients do not get time to produce antibodies already, so antigens, they are entered into large amount and they can kill a person and they can be lethal. So in that case, antibodies are injected in the form of anti-sera. The anti-sera, as I have already told you, they are it is the plasma, and anti-serum will be without blood cells and without blood clotting proteins. So anti-sera does contain antibodies, like in case of corona, the anti-sera plasma, which is called as plasma is injected into the body of critically ill patients. So this was all about immunity.